All right, hello everyone, and thanks for attending our September session of the Medical Mycology Trainee Seminar Series. Just as a reminder, the myco talks are also of interest to this community, and the next one is the 30th of September. I'll put the registration info in for the chat, and the speakers are Carl Kukla and Kirsten Nielsen, so it should be two fantastic talks, and those are free to register. So I'll pass it off to Marcia Rodriguez, who will introduce our first speaker this morning.
Thank you, Jessica. So it's great to see you all, so many friends in the audience. It's very special for me to introduce our first speaker of today, Juliana Rizzo, a brilliant scientist. Juliana is now in the Pasteur Institute, working in laboratory of Guillaume Jambon, a good friend too. And Juliana spent many years in the lab with us in Rio, and a few weeks when we moved to Curitiba. So Juliana always wanted to see the world, and she did that doing great science too. So during her PhD, she spent some time with Josh Nazanchuk in the US, and then she went to the Pasteur Institute to spend some time with Jean-Paul Atier too. She came back to Brazil, and she's now in France doing a brilliant work with fungal extracellular vesicles. Juliana has just published a great work analyzing the properties of cryptococcus vesicles. I wouldn't expect anything different because during her whole career, all that she has done was brilliant work. So Juliana, it's great to see you again. I hope you're healthy, and thanks for talking today, and they are all yours.
Thank you, Marcio, for this kind introduction. I will start sharing my slides. I hope you can all see. So I would like to first thank all the organizing committee for having me here today. It's such a great opportunity for us early researchers to present and discuss our data. So my today's talk is about the secrets of Cryptococcus extracellular vesicles. So since the beginning of my scientific journey, moving from and back to Brazil, I always kept one main question in my traveling suitcase. That was, how do cells communicate with each other? But to address this question, I also needed to address smaller ones, such as what kind of messages do they send to each other? How this message is delivered, and how by sending those messages, the cells can somehow regulate their behavior in a community level. So I came across this interpretative image during the annual meeting of ICEF, in which we can appreciate two neighbor cells, and a mailman delivering a letter to one of those cells. Turns out that this mailman is what we call today an extracellular vesicle, EV. So EVs are lipid nanoparticles delivered by all types of cells. They enclose a diverse cargo, and there's a large body of literature pointing out their role in normal physiology, but also doing pathological communication in many diseases. And here, I would like to highlight infectious diseases. So there are the two main classes of those EVs. One that we call ectosomes or microvesicles. Those are bigger particles being shed directly from the plasma membrane. And we can also see smaller particles such as exosomes being released. Those exosomes are coming from the endosomal pathway, and they are smaller particles. All those particles, they vary in terms of size, content, functions, but also depending on their source. So what do we know about EV in fungi? Although the first microscopical evidences of EV-like particles, they date from the early 70s, the first report of EVs being released by a fungal cell was only in 2007 in Cryptococcus neoformans. From this first report on, Many other reports came out, including human pathogens, plant pathogens, and also non-pathogenic species. And here I highlight Aspergillus because I also had the chance to work on EVs and cell wall remodeling when I was working on the group of Jean-Paul Latschi. So although we do know that those fungal EVs, they enclose a complex cargo comprising proteins, polysaccharides, prions, small molecules, nucleic acid, pigments. And we also know that they have been reported in more than 50 independent studies. We still have many open questions in the field. So here I list some of them. What would be their synthetic pathways and regulation, the mechanisms of EV cell wall crossing, their composition and diversity, their biomarkers, and what would be their possible functions and applications for the near future. And why studying EVs in neoformans? So we know that neoformans is an encapsulated yeast present in the environment, and they can infect humans. Those fungal cells, they can reach the central nervous system and cause the Cryptococcus meningitis. It's a very severe disease and can lead to estimated 108,000 deaths annually, especially in sub-Saharan Africa. Unfortunately, there is no vaccine clinically available against cryptococcosis or any other fungal infection. During my scientific career, I focused my research on these three main topics: the EV origin and fate, their structure and composition, and their applications. And in my today's talk, I'm going to focus on the two last aspects. I hope to come back in the future to discuss with you also my current data about the first one. So the protocol to obtain EVs is known to be laborious and time-consuming. For this work, we have used a protocol optimized by Hayes and collaborators, in which the main difference is that we collect EVs from cells growing on solid media instead of liquid media. After collecting those cells and resuspending them on our PBS, submitting them to sequential centrifugations and filtering this cell-free supernatant, we purify them by ultracentrifugation. And in the end, we have what we call an EV pellet. If we look into details by high-resolution cryo-electron microscopy, this is what we can see. A very diverse and heterogeneous population, with some particles being more elongated and other ones with a beautiful rounded shape. But the two main morphotypes that called our attention was these two. If we zoom in, we can see on the left the EVs that are completely bald and some others that were decorated by a fibrillar material. But why do the status of this decoration matter? So we do know that for fungal cells, for example, the molecules that are exposed on their surface, they are highly relevant for the host pathogen interaction. So we reason that the molecules exposed on EV surface would be also important to decipher. So we start by quantifying those EVs. So we saw that the decorated ones, they reach the majority of the population, so 89.2% were decorated compared to the non-decorated ones. We also explored them by cryo-electron tomography, in which we can here appreciate in yellow the 3D organization of this fibrillar decoration. We measure the thickness of this decoration, and the thickness could reach from 5 to 25 nanometers with an average size of 16. And we saw that there was no correlation between the decoration thickness and the diameter of these particles. Speaking about the size, we analyzed the size of those particles by two different techniques, NETA and analyzing single vesicles by the cryo-EM images. The NETA revealed to us a population of around, with a peak of distribution around 150 nanometers. And by cryo-EM, we saw that this size was shifted to the left. So the peak of distribution was smaller than 100 nanometers. This is somehow suggesting to us that the smaller population of EVs is being underexplored. And we indeed saw particles as tiny as 10 nanometers. We also analyzed the size depending on the decoration, and we saw that the decorated one seems to be bigger than the non-decorated ones. And in accordance with the previous hypothesis, we believe that at least two independent pathways are playing a role in Cryptococcus EV secretion, but this should be further explored. So when we look to the cell surface of Cryptococcus neoformans, a peculiar characteristic comes out, that is this fibrillar capsule. This fibrillar capsule is mainly composed by a polysaccharide called glucuronoxylomanan, GXM. So the most evident question for us was, would these EVs be decorated by GXM? So we purified EVs from the encapsulated and acapsular strains, and we labeled them against an antibody-raised, monoclonal antibody raised against GXM. So we could see indeed that the higher percentage of those particles were labeled by the antibody in the wild type strain, but the acapsular strain, they were not so strongly labeled. But most surprisingly, EVs coming from both strains, they were equally decorated. So this showed us that this decoration might not be GXM based. On top of that, we purified particles, EVs, from two unrelated species known to not produce GXM, Candida albicans and Saccharomyces cerevisiae. And both of those cells, they release the same decorated EVs. So this reinforces the notion that this is not GXM based. Since we found the same feature for CD neoformans and C. deuterogatiae, the two other pathogenic species in Cryptococcus genera, we reasoned if this decoration could be protein-based. And we proceed with the proteomic analysis of those EVs. So after the enrichment analysis, we found 71 EV-associated proteins, 37 being in at least two species, and 17 shared by all the three species. If we give a closer look to this list, this is what we can see. What called our attention at the first place was that 10 out of those 17 were predicted to be GPI-anchored proteins, and some of them play important role in cell wall metabolism, such as glucosidases and glucosyltransferases. On top of that, if we look at the protein family levels, we could see that the three members of the chitin deacetylase family, CDA1, 2, and 3, were all present in those EVs from the three species. On top of that, we could obtain EVs enriched for the families RIL1, GOX, RIL, GOX, and also it's important to mention that we found the protein family of tetraspanin, that it's known to be a marker for mammalian-derived EVs and also suggested as a marker for Candida albicans EVs. I also wanted to highlight that the most abundant protein across all the species, all the three species, was the monoprotein 8. And not only this one was found, we found also MEP98 and MEP84. So in face of that, and also taking into account that the outer layer of the cell wall of many fungal pathogens is composed by manosylated protein, we reasoned that the decoration of those EVs could be specifically monoprotein-based. To address that, we obtain EVs from the encapsulated and acapsular strain. We label them with concanavalin A, that is binding to the monosyl group of those proteins. And after ultracentrifuge washing, we analyze them by flow cytometry. And this is what we could see. Regardless the cellular source, the EV population were similarly labeled by CONE and strongly labeled. In addition, we treated those EVs with proteinase K, and after blocking the enzymatic reaction, we labeled them with CONE, ultracentrifuge wash, and analyze them by flow cytometry. And again, regardless the cell source, EVs lost their cone labeling after the protein with the treatment with proteinase K. So we then reasoned that the deletion of some genes related to the protein glycosylation pathways in Cryptococcus could provide us some insights about this decoration. So we used some mutants defected in N-linked or O-linked glycosylation, such as ILG3, KTR3, and HOG3. And we also created mutants on the two most abundant proteins in neoformans EVs, MEP88 and the protein that we call VEP1. So if we look into the EV production, we saw no big differences among the strains, but MEP88 seems to be slightly increasing the EV production. When we look at the the percentage of cone labeling, we could see that MEP88 again and the double mutant MEP88 VEP1 were producing EVs that were class labeled by cone. If we look into the details by cryo-EM, we did not see any difference in terms of EV size, but we did see a difference regarding the decoration thickness. So for ILG3 and MEP88, we saw a decoration thickness that was less dense compared to the wild type, as you can see in your right panel. So with that, we conclude that this decoration is probably formed from a dynamic combination of monoproteins, since none of our mutants completely abolish this feature. But you might ask, do you believe that all fungal-derived EVs are decorated? And my guess is no, because if we look back into the literature, we could see that the Malassezia sympodialis derived EVs called MALA-X, when analyzed by cryo-EM, they do not seem to have a very evident decoration. And this is in accordance with what we know about the cell wall of this fungal pathogen. So Malassezia sympodialis is known to lack the N-glycosylation pathway. They have also a small number of GPI-anchored proteins, and the fibrillar layer of the outer cell wall is not the same. So this just highlights how diverse fungal EV are. And with that, we propose a new model of Cryptococcus EVs, in which the lipid bilayer is covered by GXM as the outer layer, and by this fibrillar structure composed by many GPI and transmembrane proteins. We could see, and what called our attention was that many of those proteins, such as the family of COX proteins, CD8 and MEP88, was previously described as immunogenic and even tested as vaccine candidates. So we reasoned that EVs could be a platform for immunization against cryptococcus. And to test that, our vaccination strategy was based on immunization of Baubse female mice with CAP59, so they are capsular mutant-derived EVs. Our control group consisted of mice injected only with PBS. So after immunization, the serum was collected and were blotted against the EV components. So here by Western blot, we can see for each lane the serum from individual mice that was blotted against EV components. And we could see that all immunized mice produced antibodies against EV components. And this was not the same for the control group of mice. It was completely negative. Once the antibody response was confirmed, we then proceed with the infection of those mice with the wild type strain. So we challenged them intranasally with the wild type and monitor their survival. So what we could see was that all immunized mice could survive longer than the control group. But whether the protection was mediated by adaptive, innate immunity, or even trained immunity needs to be further explored. So as summary, we can see that Cryptococcus EVs are more complex than previous thought. And we believe that this might be also true for other fungal pathogens, as outlined in our recent review. And speaking more specifically about Cryptococcus EVs, we know that they bear a monoprotein-based decoration, that this decoration is mostly made by transmembrane and GPI-anchored proteins as their hallmarks, and they have a strong potential use as a vaccine platform against fungal infections. So as future perspectives of this specific work, we aim to separate these two subpopulations of decorated and non-decorated EVs to characterize them in terms of proteins and RNA content, and also maybe modify the antigens exposed on EV surface in order to increase the EV protective effect. Beside that, my interests are currently based on a screening of a mutant library in which we could find 4 good candidates to move forward. We are also interested to optimize an intracellular communication protocol to try to further address how EV-mediated communication can somehow be also involved in drug-resistant mechanisms. With that, I would like to thank all my mentors, Márcio Rodrigues, Joshua Nozanchuk, Annie Bouvet, Jean-Paul Attier, and especially Dr. Guillain Jambon, to be extremely supportive during my scientific career. I also would like to thank my lab mates, especially Frédéric Moirin, that helped me a lot during this project, all the collaborators, of course, the funding agencies, and I especially would like to thank you all for your attention, and I will be happy to take any questions. Thank you very much.
So we're taking questions after the second speaker, right? So Jessica, if you want to take control.
Great. Thank you, Juliana, for a wonderful talk. And now I'm very pleased to introduce our second speaker, Thierry Rolling. And Thierry received his MD from the University of Freiburg, and then received extensive training and experience in both clinical and experimental science. These include a Master's of Science at the London School of Hygiene and Tropical Medicine, clinical research fellow positions in Gabon and Hamburg, and a residency and further clinical training at the Universität Klinikum Hamburg-Eppendorf. In 2019, he joined Tobias Holz's lab at Memorial Sloan Kettering Cancer Center and is investigating the microbiome in terms of both fungi and bacteria. And today he will talk to us about his microbiome work. Thierry?
Yeah, many thanks for this kind introduction and possibility to share my work with you. So yeah, I'm going to talk about what I did in the last 2.5 years here in New York and present the data on the microbiome in patients undergoing hematopoietic stem cell transplantation. And okay, it doesn't move forward. No, it does. Okay. So just a brief introduction about what is hematopoietic cell transplantation. Basically, it's a curative treatment for certain malignant and non-malignant diseases of the blood system, such as leukemias or lymphomas or other cancers of the blood system, basically. And what is HCT? So basically, you do a conditioning regimen where you either give the patient body irradiation or a high dose of chemotherapy, basically to delete basically all the stem cell and the blood cells in the patient. So basically replace the cancerous blood cells with healthy donor cells in these patients. But while this is a curative treatment, unfortunately, there are complications associated with this process. So the first one is like due to the high-dose chemotherapy and the immunodeficiency resulting thereof, the patients are at a high risk of opportunistic infections, bacterial infections, but also fungal infections. And the second one is that the donor hematopoietic stem cells actually can attack the patient, the recipient, because of slight mismatches. And this T-cell driven response leads to an inflammation called graft versus host disease, which is basically an inflammation mostly of the gut or of the skin in the patients. There's actually a lot of literature already, or a lot that we know about the bacterial microbiota and patient outcomes in hematopoietic cell transplantation. And a lot of this data has actually been produced here at Memorial Sloan Kettering Cancer Center. So we know that patients that receive broad spectrum antibiotics have worse outcomes. We know that patients that have a low bacterial diversity in their gut also have worse outcomes. And actually, we can point it down to specific bacteria. So patients that have an expansion of enterococci, for example, have worse overall outcomes and a higher risk of infections with enterococci. And this has been reproduced in multiple studies in different centers. So there's really a strong link between a bacterial dysbiosis of the gut and increased risk of infection, increased risk of graft versus host disease, and lower survival in patients undergoing stem cell transplantation. So the big question for us is, what about fungi? Do fungi play a role in the gut in hematopoietic cell transplantations? And in my work, I look at a cohort of 156 patients undergoing stem cell transplantation. And this is a fairly standard representation of the population here at Memorial Sloan Kettering Cancer Center undergoing stem cell transplantation. So a median age of 60, and most patients received a stem cell transplantation due to an acute leukemia and myelodysplastic syndrome, lymphomas as the second and third most common indication for this. And of these 156 patients, we have 1,279 fecal samples collected between 10 days prior to the transplantation until 1 month after the transplantation. And we did basically a multi-omics approach with this. All the samples, we cultured them for fungi to see if there are viable fungi in the gut or in the feces. We did fungal ITS1 sequencing, and we also did concurrent bacterial sequencing in qPCR to check for interactions between the bacterial and the fungal microbiome in these patients. So first, we looked at overall dynamics of the microbiome and the mycobiome. So on the left, we have the data for the fungi, and on the right, we have the data for bacteria. And these are like fairly standard values that you look at in microbiome research. So on the top, we have the number of fungal DNA, 18S copies, basically, and the same for bacterial, 16S copies per gram of stool. And you already see there's a difference between fungi and bacteria. There's the fungal 18S copies, there's a lot of variation in there, but basically there's not a lot of change between pre-transplant and post-transplant. Statistically, the copy numbers are a bit higher after transplant, but if you look at it, the difference is really small. While for bacteria, there's really a trend that before transplant, they have relatively higher 16S copy numbers, but then the number of bacteria declines throughout the transplantation period. You have to, this is log scale, so this is quite a huge difference. And this is, of course, due to patients getting antibiotics, so the bacterial load really decreases. And a similar picture is seen if you look at the diversity. So the first global measure for microbiota, which looks at like how many different species of fungi or bacteria in there, and how often they are there, how their relative balance is there, basically. And again, for the fungal diversity, it's really stable. There's not a global trend over the whole course of stem cell transplantation. While for the bacteria, there's a striking decrease in bacterial diversity. So higher diversity, like a normal gut flora before the transplantation, and then patients have like a dysbiosis after transplantation with very low bacterial diversity, which has been shown. So there's really a striking difference between the dynamics of the fungal and the bacterial microbiota in these patients. And we can quantify that if we look at the similarity of the fungal or the bacterial microbiota between two samples. So this is a bit complicated data, but I'll try to explain. So each data point here is basically the similarity between two individual fecal samples. And the similarity measures, this is measured by a Chow-Jakar distance for those who are interested in it, but basically a number of 1, and this means that the sample from one day and from the other day is exactly similar, like if they are completely different, so if completely different species are in there, the similarity would be 0. And if like part of it is in day 1, but only part of it in day 2, there's the similarity measure is somewhere in between 1 and 0. And then basically the first ones, that's all samples that are apart by 1 day, then and so on. So the last ones that are 2 sample pairs that are away by 30 days. And I'll start on the right with the bacteria. This is something that's known. So there's basically a power curve decline in the similarity between samples for the bacterial microbiota. So if you have samples that are collected 1 day apart, for the bacterial microbiota, they are pretty similar. But the longer you have between 2 samples, the less similar they are. But so there's a gradual change in the bacterial microbiota, but not from day to day. But for the fungi, there's actually like all over the place, there's a high variability. So you have some samples that are completely similar, some samples that are completely different, and there's no real global decline over the time period. And especially like day-to-day variation is much higher for the fungal microbiota than for the bacterial microbiota. Now, what could the reasons be for this? I mean, we know that a lot of the bacteria that we actually detect and the DNA in the stool samples actually are colonized the gut, but for the fungi, we don't really know. And some of the fungi that we actually detect is not from colonization, but rather from transient passage. Because this is like an interesting work done in the group of Joe Petrosino, which shows that basically, for example, Saccharomyces cerevisiae, you see it in the gut ITS data only if patients have their regular diet, but if you remove like bread from their diet, they have much lower Saccharomyces cerevisiae. Similarly, in this work, they could show that if patients have a better oral hygiene, like brush their teeth more often, the relative abundance of Candida in the fecal samples decreases. So a lot of the fecal DNA we detect in the gut probably is just transient and from environmental factors. And that would explain why there's a higher day-to-day variation, because it's not really colonization, it's just like going through the gut. But nevertheless, there's something interesting we found in our patients, and so this is not an abstract art, but this is like an overview of all the samples in our cohort. So 1,279 samples with fungal amplicon sequencing data in there, and the different colors represent the relative abundance of specific fungal taxa. We see the more common colors are here, that's Candida albicans, then in yellow it's all Candida parapsilosis complex, and here in purple it's Saccharomyces cerevisiae. So those yeasts are the ones we see the most common, very high relative abundance in our patients. And then when we compare this below, like in black, it means that the specific sample also had a positive fungal culture when we cultured the specific sample. And you can already see that there's a cluster near yellow samples, so a cluster in samples where Candida parapsilosis dominates the relative fungal abundance. And actually what we can see is that while there's a lot of variation overall in the microbiota from day to day, some patients exhibit a really stable expansion of vital Candida parapsilosis in the gut. So you see here, pre-transplant, there's variation, there's some aspergillus here, this is Saccharomyces, and then they have like almost 100% of Candida parapsilosis sequences, amplicon sequences in their fecal samples, and at the same time we can culture Candida parapsilosis at high levels, similar for these patients, while other patients, they have a more variable microbiota, and we cannot culture fungi from these. So for further analysis, we decided to call domination event a sample in which 90% of the sequences are from one specific fungal taxon. And then if we compare the similarity, so basically if I have a sample today, how does it look tomorrow? We see that samples which have fungal domination, so in which 90% of the sequences are from one single fungal taxon, the sample from tomorrow is much more similar than the one from today. And this is mostly driven by Candida parapsilosis complex domination. So basically, if a patient is dominated by Candida parapsilosis complex today, he probably also has a domination tomorrow. So what this means, like before I said, there's much day-to-day variation in the microbiota, but not if you have Candida parapsilosis domination. Then there's a stable phenotype where the patient is really colonized over several days by Candida parapsilosis. So this is very intriguing and interesting to see. And similarly, this confirms the data. So if we look at samples in which there was a domination, so basically there were 59 samples where there was Candida albicans domination, so 90% of the sequences were from Candida albicans. There were 54 of Candida parapsilosis domination, etc. We see that actually only the Candida parapsilosis complex dominated samples also are almost all, or to a very high degree, culture positive. So what we can show is that Candida parapsilosis domination in these patients is actually a stable colonization and associated with viable fungi being retrieved from the fecal samples. So while the others, we can't culture the fungi as often from this gut. So basically, it's more likely that it's from some environmental source and just not real colonization in these patients. And if we look at the time course, we see that Candida parapsilosis complex domination mainly occurs like in the first 2 weeks after transplantation. And similarly, culture positivity is much higher post-transplant and pre-transplant, which is in line with culture positivity being associated with Candida parapsilosis domination. So you could ask, why do we see this Candida parapsilosis complex expansion, and why can we culture it while we can't see it for Candida albicans, for example? And this is the data by Bing, who is also in the audience. So one of the reasons probably is that in our center, we use to prevent invasive fungal disease, all patients receive micafungin prophylaxis. And then it is switched to an azole after 7 to 10 days after the transplant. And actually, Candida parapsilosis has a slightly higher tolerance to micafungin than Candida albicans. So that would explain why we see it. And actually, Bing did a mouse experiment where basically mice were gavaged with equal amounts of Candida albicans and parapsilosis. And then either they received micafungin in the darker shade here, or they didn't receive micafungin. And if the mice are given micafungin, there's basically Candida parapsilosis it dominates. Basically, you kill the Candida albicans, but Candida parapsilosis is still there. So that would explain the phenotype also we see in our patients.
Hi,
now,
my name is Sophia Campuzano, and I recently got married. And
hello? Okay. I'll just continue. So basically, and then the next question was what's the interaction between the bacterial microbiota and the fungal microbiota in our patients. So we compare samples which had no fungal domination to samples that had Candida parapsilosis, Candida albicans, and Saccharomyces cerevisiae domination. So over 90% of the amplicon sequences we detected were relative to these species. And we can see that Candida parapsilosis complex domination is associated with an almost 100-fold reduction in the bacterial load. So we really see that patients that have a low bacterial biomass are more likely to have an expansion of Candida parapsilosis. So, and we also looked at like specific bacterial signatures associated with an increase in Candida parapsilosis albicans or cerevisiae. And we basically see that there are different signatures that we see here. I'll concentrate on the Candida parapsilosis ones. So an increase in Candida parapsilosis complexes normally correlates with an increase also in Staphylococci, for example, or some other fungus species, bacterial species, but families. And the interesting thing about it is that, for example, Staphylococci and some of the other positive correlations that we see here are normally not like the typical bacterial flora of the gut. And what we see is that we see an increase in Staphylococci relative abundance also in the context of decreased overall bacterial abundance. So basically this sum is in line with the data that if you have low bacterial load, then Candida parapsilosis can really expand in the gut. An interesting thing we also see is that if Candida parapsilosis increases, there's a decrease in Enterococci. which independently has been shown to have worse outcomes in the patients. So that's for the descriptive part of the bacterial and fungal microbiota. The next thing is like, well, does it even matter if Candida parapsilosis expands in this patient? And yes, it does matter. It's actually really bad for the patient. And this is Bing's data that has been published last year, and she talked about, I think, a month ago in this seminar. So I won't go into the detail, but she show basically that patients that have an expansion in Candida parapsilosis complex species are at a higher risk of having a Candida parapsilosis complex bloodstream infection. So if you have Candida parapsilosis expansion in your gut, then it can translocate to the bloodstream and cause bloodstream infections. And for my work, I expanded this to other adverse outcomes in the patients, and I looked at overall survival. And I can actually show that patients that have Candida parapsilosis complex domination in their gut around the time point of transplantation have a much lower survival than patients without Candida parapsilosis complex domination. And this is independent of fungal bloodstream infections because none of the patients with Candida parapsilosis complex domination in the gut that died here died of a fungal infection. So it's really irrespective of the bloodstream infection. And if you go into detail, you can also look at what are the causes of death in these patients. And the major category why people die when they have a Candida parapsilosis complex domination is called transplant-related mortality. So that's basically all kinds of complications that could be graft-versus-host disease, that could be other complications of the treatment the patients had, that can be infection. But most importantly, it's not due to the primary disease of the patient. So it's not recurrence of their leukemia or whatever. It's really related to the transplantation. Then we looked at whether this is really holds true also in a multivariate model. If we correct for factors that we know that influence outcomes, so older patients are known to have a lower survival after stem cell transplantation. Patients with a higher risk score have a higher risk of dying after transplantation. And as I showed earlier, like patients that receive antibiotics such as carbapenems or piperacillin tazobactam have a lower survival. So we corrected for all this, and even after adjusting for all these factors, Candida parapsilosis domination is still associated with a worse outcome in the patients. So to summarize them, I can show that Candida parapsilosis can expand in the context of a low bacterial biomass in these patients and under mycofungin pressure. And basically, Candida parapsilosis expansion is bad for these patients because it can be a source of bloodstream infections, and it's associated with a lower overall survival. And with that, I want to thank everybody in Tobias' lab, especially Tobias, and then also Bing, who unfortunately left our lab a few months back. And also a big thanks to Mergim, who helped a lot with preparing the 1,279 samples. Also a big thanks to other people at MSK that are involved in the microbiota research, and of course to the funders, especially the German Science Foundation that funded my fellowship here in Tobias' lab. And then finally, if you're interested in the fungal microbiota or in antifungal immunity, the whole lab is hiring. So yeah, if you want to do awesome science in an awesome city, you should join our lab. Many thanks.
All right, thank you, Thierry, for a wonderful talk. If everyone would please enter questions into the chat, we will go back and forth between the speakers, but to give people time to enter questions for Thierry, we're going to start with questions for Juliana. Marcio?
All right, those were two great talks. So it was a nice session. We are starting with Juliana taking 3 or 4 questions. Then we are going to give some time to Thierry, and then we can come back to Juliana, right? So Juliana, you have questions about the treatment of antifungal cell, of the fungal cells with drugs, and then EV production from Anna. You have a question from Sandeep asking if you have checked the localization of the GPI proteins in the EVs. And Matthew is asking about the RNA content in crypto EVs in the biogenesis too. And Jessica is asking about the effect of the vesicles on the host cells and maybe a relationship with the RNA too. So we're starting with these 4 questions. If you want to combine your answers, it's okay. If you want to go through them individually, it's okay too. It's just about the way you want to answer.
Thank you, Marcio. I will start with the biogenesis. So in terms of how EVs are produced and if we have tested or not different drugs and analyzed their production. So far, we are mostly working with fluconazole. And we do see that these drugs can indeed affect the EV production, but we do know that this EV production is highly also dependent on the media. So we were quite surprised when we saw that the yield of EVs was much higher on solid media in comparison to liquid media. And we are now testing different culture conditions. And the amount of EVs that we get is highly different between one condition and another. And also during antifungal treatment, yes, they are, let's say, they are kind of modulated by the growth conditions. In terms of the biogenesis, also related to GPI-anchored proteins. So far, we haven't tested any of those proteins specifically in the EVs, and we still do not know if this is a mechanism of loading those proteins inside the EVs, or if somehow these proteins could be taken from the cell wall once the EVs are crossing the cell wall. So this is something that we aim to do, to look into the details of those proteins in the EVs themselves. But so far we haven't tested. So these are predicted GPI-anchored proteins, and we need to further explore. About their function in terms of interaction with host cells, there's something on the EV field that we call paradoxal effect. So in some conditions, EVs can be protective, but in some others, so there are some papers on Sporotrichis, for example, that these EVs can be harmful for the host and can be bad for the progression of the disease. So I think it highly depends on the molecules that are exposed. So it's mostly how this message is delivered. So for each condition and each way that the vesicles are obtained, this can make a big difference on the function that we explore. So
I would say that the amount of vesicles makes a difference too, because we don't know how many of them are produced during infection. So I think it could change everything. So, all right. So Jessica.
All right, thank you. A question from Kyla Ost says, great work. Is reduced transplant survival associated with domination by other fungi, or is this effect specific to Candida parallelopsis?
So, well, I mean, I can't answer that from our data, but so as I said, due to the specifics of using mycofungin prophylaxis, we probably select for Candida parapsilosis, and that's the one we see colonizing our patients and leading to adverse outcomes. So in our data set, I looked at Candida albicans domination and other Saccharomyces domination. Basically, that has no effect on the outcomes of our patients. But now if you have a different center that uses, or if you use a different antifungal prophylaxis, or if there's a different epidemiology of fungi and in the center's patients, of course, it's possible that this is not specific to Candida parapsilosis, that maybe if you have colonization by Candida albicans and expansion in the gut in the post-transplant period, you could also have a similar effect on survival. So my hypothesis is that it's not Candida parapsilosis specific, but that we only see it here because of local epidemiology. But I can't really prove that for now. So.
Right, that leads into the next question from Sadri Zanati, which is, knowing that mycofungin selects for this expansion to parapsilosis, what would this mean from a perspective of antifungal stewardship?
Yeah, so basically, so I mean, the initial clinical trials on antifungal prophylaxis were with azoles and with fluconazole. So we have a good evidence base actually to use azoles in preventing invasive fungal disease in these patients. But then the azoles have a higher interaction potential, especially with chemotherapeutic drugs and calcineurin inhibitors that are used by the transplant doctors. So that's why some prefer to use an echinocandin in the first few days until basically the immunosuppressive drugs have reached a steady state. So that's basically a discussion you have to have locally with the hematologist, oncologist to discuss what's the best way. But from an infectious disease standpoint, I personally would say it's better to use an azole. But that's something you have to discuss because you're not the only ones that are treating the patients. So.
All right, why don't we send it back to Marcio for questions for Juliana?
Thank you, Jessica. So, Juliana, we have two related questions from Vikas and Thierry related to the conditions at which vesicles are produced and the impact that these conditions could have on the composition and morphology. So, and then we have a last question from Giuseppe, a very tough one, I think, related to cell wall remodeling and docking of EVs.
So in terms of the origin sites of those EVs, so we believe, as I mentioned in my talk, that we might have two pathways playing a role since we found those two specific morphotypes. But we still don't know. That's why the genetic screening that we are doing so far, it's highly important to review the genetics and the transcriptomics behind the EV production. But we do know, for example, that those EVs, they enclose many molecules such as mRNA, different subclasses of non-coding RNAs, and they are involved in cross-kingdom communication. There is even a paper on the plant, fungal plant interaction in which the EVs can somehow silence the expression of some virulence-related genes. So we have some very strong suggestions that those EVs, no matter where and how they are produced, they are playing a role in the intercellular communication. But so, for example, during cell wall remodeling, it's have already been shown for Saccharomyces cerevisiae and also in our previous paper on Aspergillus fumigatus that when the cells are, so when I obtain protoplasm from Aspergillus cells and let them regenerate the cell wall, the components of those vesicles and the amount of vesicles that are recovered in the supernatant is completely different. So we still need to have stronger arguments about cell wall remodeling, but there are already nice indicatives in the literature that they might play a role. And the fact that they have those enzymes, like we still need to prove that these enzymes are active on the EVs, but the chitin deacetylase, for example, this is highly suggestive of cell wall remodeling. So I don't know if I answer, but
yeah, thank you, Juliana. We can go back to Thierry.
All right, so I have two questions, host prerogative, and that is, so you showed the very nice data about how bacteria can be positively and negatively correlated with prolapsosis levels. And so I was wondering whether the species that are negatively correlated might be producing some antifungal compound. Would it be possible to exploit that?
Sure. That would of course be great if we could find that out. That's one possibility, of course. But I think that's something we have to look at in more detail, like in mouse experiments and like mechanistically expand, because this is, for now, it's correlations in the human data. So, but that would be great. Yeah.
Yeah, and then. I thought your work really nicely demonstrates the importance of antifungal tolerance. And so what do you think about this clinical, do you think that the tolerance is clinically important? And how should we think about tolerance versus resistance when thinking about antifungal treatments?
Okay, that's more difficult to talk about. So basically, like the most of the Candida parapsilosis would still be classified as susceptible, but it's like it has the minimum inhibitory concentration is a bit higher than for Candida albicans. So that's why if you give a drug, you select for the ones with the higher. So that's just an intrinsic factor. Like, but again, like if you have a bloodstream infection with Candida parapsilosis, the risk of a treatment failure with niclonacandin is slightly higher than if you have with Candida albicans. So overall, I think like treating fungal infections and fungal bloodstream infections is more complicated than treating bacterial infections, and especially the antifungal susceptibility testing that we got, the correlation to clinical outcomes is not as good as for bacteria. So there's definitely room for improvement to understand tolerance and resistance mechanisms in these in fungi, and also to see whether this actually translates to differences in patient outcomes. So.
All right, those are all the questions I see in the chat. So if you have any questions, feel free to turn on your mic. But otherwise, thank you to both our speakers for fantastic talks. And thank you everyone for attending.
Thanks.
Thank you.
Right. Yeah, those were great. I really enjoyed them. So thank you both very much.
Yeah, thanks for having us. Bye.
Very nice science. Congratulations.
Mm-hmm.